So thank you very much. Thank you for the kind introduction. And so my name is Kevin Rupp. Hi. And I'm a PhD student in the lab of Nico Bernwinkel in Switzerland. And it's a really great pleasure to be here today and to talk to you about modeling metastatic progression from cross-sectional cancer genomics data. So before I talk about our method, I'd like to give you some intuition about the metastatic disease or metastatic process from a genomics point of view. Specifically, I'm talking from a bulk genomics point of view. So I'm not going to talk about intratumor heterogeneity. Assume that the metastasis starts always with a healthy patient. And a healthy patient is being characterized by the complete absence of any pathogenic mutations or cancer driver mutations for that. Then, due to random processes over time, some of the cells in this patient's lung get a pathogenic mutation, in this case KRAS. This gives them a proliferative advantage so that the cells are able to outcompete the normal surrounding cells and start growing faster. So, over time, they form a tumor, and the tumor grows and grows and itself accumulates mutation. Not all of them confer an advantage, but those that do can have a wide variety of advantages. For example, some of them might confer a migratory advantage, so that cells with that genotype are more motile, meaning that they can invade the surrounding tissue more easily, or the lymph nodes, or the vasculature. And some of the cells that make it to the vasculature might travel through it and leave it at a very distant point in the patient's body. For example, here some cells make it to the brain, and there they're able to form a new colony. This colony is known as a metastasis, and this migratory process can be called metastatic seeding. At the start, the metastasis is a copy of the primary tumor's genotype. However, due to random chance and due to different selective pressures at the distant site, it might acquire a very different genotype from their primary tumor. So their genotypic profiles start to diverge. Both grow at their own pace, and at some point in time, the primary tumor is so big that it causes a patient pain. So he seeks medical attention and gets the primary tumor diagnosed. However, the big problem during metastatic progression is that the metastasis might not yet be diagnosable when the primary tumor is. And so it remains in the patient's body for a longer amount of time. And it grows, it evolves, until itself becomes diagnosable. So, unfortunately, we do not observe this metastatic process directly. What we do observe are its endpoints, meaning the genotype of the primary tumor and the genotype of the metastasis at the time of the respective diagnosis. We do observe this not only from a single patient, but rather from a whole cohort of patients. And from there, we can then investigate trends among them, some patterns, for example, like mutational co-occurrence, or some enrichment of metastasis of mutations in metastasizing tumors versus non-metastasizing tumors. And our task now as computational biologists is to infer a model from this kind of data that explains these patterns. And so here I'd like to introduce you to our mechanistic model that we call MET-MHN, that we fit on this data and use it to reconstruct the most likely orderings for each observed data points to better understand the process of metastatic progression and initiation. So, speaking about MET-MHN, it's called Mutual Hazard Networks for Metastatic Disease, and it's a member of the MHN family of models. All of them are cancer progression models that model the progression of cancer as the accumulation of different genomic events. All of them do this as a continuous time Markov chain on a binary state space. Specifically for MET-MHN, our states, I guess somebody scored a goal. Specifically for MET-MHN, our states is a pair of genotypes, that of the primary tumor and its metastasis. We model each event by 2 bits, where the first bit encodes the presence or absence of the mutation in the primary tumor, and the second bit does the same thing in the metastasis. We model both copy number alterations as well as mutations, and additionally, we are also modeling the diagnosis of a tumor as a distinct event itself. The only event that we do not model by 2 bits, but rather by 1 bit only, is the so-called seeding event. Here we define the seeding event to be the point in time when the primary tumor and the metastasis start to branch away from each other. This can also be seen in the state space. So some notes about that. We assume that all mutations accumulate one by one and irreversibly, meaning once a mutation has fixated, it cannot be lost again. Then, for MetMHN specifically, we assume that prior to the onset of the seeding event, the primary tumor and the soon-to-be metastasis still form one physical entity, meaning that each mutation that accumulates in the primary tumor has to accumulate in the soon-to-be metastasis at the same time. This leads to a very sparsely connected state space. All of the transitions on that state space, we parameterize the rates according to the mutual hazards parameterization, meaning that we split them up into a product. In a product of base rates, this is the rate with which a mutation accumulates on its own or spontaneously, times the multiplicative effects of all mutations that are already present in that tumor. So these multiplicative effects, you can think of them as epistasis, for example. And exactly these base rates and these effects between mutations are what we want to infer from data. These will form our mutual hazard networks. By construction, the last event that happens jointly in the primary tumor and the metastasis is the seeding event, after which these two sequences spread apart from each other. And they start to accumulate mutations independently from one another on their own. And additionally, the metastasis might be in a very distant tissue. We account for the foreign selective pressures that this new mutant now faces by an additional factor, namely the influence of the new tissue on the rate of fixation on all mutations. So we also model the observation of a tumor as a direct event. And why do we do this? Mainly because we're only working with observational data. All of our clinical genomic datasets at hand are purely observational, meaning that each patient that has a tumor and appears there has to have been diagnosed. And only the patients that have symptoms do get diagnosed. So the data generating process is inherently biased or even conditioned on the tumor having shown symptoms. And in order to overcome observation biases from this, we adopt the technique that was being introduced by Schill et al. This year and model the tumor observation as a direct event itself. And the observation of a tumor directly depends on the genotype of that tumor. So all states can be diagnosed, or all tumors can be diagnosed. And once the diagnosis happens, we assume that no further progression happens in that tumor. You can think of this as the tumor being biopsied and the biopsy being physically frozen away. There's nothing happening in that anymore. From a Markov chain point of view, or theory point of view, all of these states are absorbing, or probability sink states. And this construction is quite handy for us when we're trying to come up with probabilities distributions over that space. So in Markov chains, we're mainly interested in modeling the transient distribution of a system. And for the transient distribution, we need 3 ingredients. We need a model that I already described to you. We need a starting condition that you see here, where we assume that the entire probability mass lies on the healthy state, on the wild type state. And we need a notion of time. In this case, it would be the amount of time that has passed from tumor initiation to its diagnosis. And this information will always be hidden for us. It's not something that we can physically measure in any way. And so, in order to circumvent this, in order to get rid of it, we're not working with the transient distribution, but rather with the stationary distribution. And from the theory of Markov chains, we know that the entire probability mass of a stationary distribution has to be located on absorbing states. And by construction, all our absorbing states are the states where an observation has happened. So effectively, we're modeling the distribution over potential tumor states at the time of first diagnosis. So whenever there's a first diagnosis, there might be another diagnosis. And this is the case for many metastatic tumors. We first observe the primary tumor many years before we observe the metastasis. We account for this. So as soon as we get an observation of the primary tumor, we take the distribution and condition it on that observation by selecting the set of states where the primary tumor genotype matches the observed genotype. Then we set the probability mass of all our states to 0 and renormalize. Then in the next step, we propagate this distribution forward in time in a mechanism that's very similar to the one that I just described to you. And then we can evaluate the probability mass of our full observed state as soon as we get that observation. So we can give a probability for each tumor state, and with that we can define a likelihood and fit that in a maximum likelihood manner to data. And the data we chose for this study at first was the lung adenocarcinoma data from the MSK impact study. And this data consists on this clinical panel data and its single region sequencing. We have around 5,000 patients overall, where around 450 of them have both the primary tumor and the metastasis sequence available. We choose a panel of 28 genomic events, plus the seeding event, and of course 2 observation events. And here you can see the main output of our method, and that's the MHN. And the way to read it is as follows. Here in the rows, you see the causes, so mutations, for example, and in the columns, you see the effects of these causes on all other events, meaning those are the effects of mutations on the rate of accumulation on all other mutations. In the first column, you see the base rates of with which these mutations occur. And a quick note, everything is plotted here on a log scale. So let's zoom in a bit to discuss it further. And there I'd like you to focus on this edge between EGFR and KRAS. Both have a very similar negative effect between those, meaning that if one of these mutations accumulates, then the other mutation will likely not accumulate at all, meaning this is some sort of antagonistic effect between those two. If you exponentiate the effects, then you'll find that KRAS decelerates the rate of accumulation of EGFR by a factor of 36, meaning it's much more likely that the tumor gets diagnosed before EGFR can hit if KRAS is already present. And this is something that's very well described in the literature. Another effect pair that's very well described in the literature are the effects between STIC11 and KEAP1, where we find a positive synergistic effect between those two. And it's not only positive synergistic, but what we also find is that it has a directionality to it. The effect of STIC11 on KEAP1 is much larger than the other effect, or vice versa, which we can also find evidence for in the literature. We can not only find effects that are present in the literature, but our model also is able to suggest effects that have not been validated yet. So it can generate hypotheses that we can test in the lab. But what interests us most are the effects of mutations on the rate of seeding or on the metastatic potential. And there we find that EGFR has the biggest impact on the overall rate of metastatic outcomes. There's also some evidence in the literature which might corroborate this. But it's not just EGFR, but rather a lot of cell growth drivers in the RTK-RAS pathway, including BRAF and MET as well. Then we asked, what's like a general model, or what are general timing trends here? And for this, we calculated for every mutation the probability that it appeared the distribution of its arrival time. So when it happens, when does this mutation happen, given that it happens? And there we see that several mutations, such as KRAS, tend to happen rather early on. So meaning we see some definitive set of initiating primary drivers, as well as some typical seconds and thirds that do not have a strong enough impact to initiate initial growth. But if an initial driver is there, then they accelerate tumor progression. The metastatic seeding is also such a similar event. It happens towards the middle of primary tumor progression. Interestingly, we find that copy number alterations tend to happen rather late during a progression trajectory. And this is not just some general trend, but we do also observe in patient samples. So for many patients, we reconstruct the most likely trajectories. And here, just by visually inspecting it, you see a lot of heterogeneity going on between the patients. So patient 1 has a lot of mutations accumulating prior to the seeding. Patient 2 has EGFR and a copy number alteration, which on average tends to happen late. And patient 3 only needs a single mutation. What I want to highlight here is that our model does not make any parsimony assumptions. That you can see here, that it puts this event, a deletion in TP53, in a non-parsimonious way on that trajectory. It estimates that in the most likely trajectory, these events happen twice independently from one another rather than once early. So, and with this, I think I'm towards the end of my talk, and I'd like to take the opportunity to thank everybody who was present during this process. And especially I'd like to thank my supervisor, Nico Bernwinkel, my former master student, Sheng-Shi Ni, my long-term friend and supervisor, somewhat supervisor, I guess, Rudolf Schill, and also our collaborators from the University of Regensburg, specifically Linda Hu and Andy Lösch and my former mentor, Rainer Spang, who are both here also today. Thank you to all of you, and also thank you to you for listening. And should you be should you be interested in this project, here's the link to our GitHub and also the link to our paper. Thank you.
Thank you. Questions? Everybody is taking pictures of your links.
Hi, great talk. Thank you for sharing. I was wondering if you had taken a look at comparing your model of progression of these genes with, say, like the TracerX consortium results in lung cancer.
So we did some comparisons, not specifically with TracerX, but rather also with TCGA, because we had the data more easily available, and we could still validate these trajectories on the primary tumors. And there we took a look at them, and we compared these trajectories or these lines of descent from phylogenetic models with ours, and found that there's definitely some overlap, especially in the driver mutations, but they do tend to sometimes not agree on certain pairings.
Okay, thanks.
Thank you.
A great talk. Actually, in these kind of studies, when you consider paired, primary, and metastatic, treatment has to be accounted for, right? There are in lung cancer, there are at least 3 different subtypes, and subtypes are treated differently. So yeah, the chemotherapy is a carcinogen, it's bound to make mutations. So how do you account for that? How do you control this covariate?
Very good point. And our answer has to be not at all. Because the sad reality is that we do not simply have this data available for the data set we're using it. Only a very small cohort of those is annotated. And I guess the bulk of that is still lacking this information.
At least, will you be considering the subtype information?
So for the subtypes, we had only this non-small lung cancers and only learned on those. So we did not utilize any other tumor types in that.
Okay, in the future, hope. Thank you.
Thank you.
Okay, we will need to move